Dear students, let us discuss another topic uh, from the second unit of your syllabus of the zoology in semester 4th, which is linkage and its types, incomplete linkage and the linkage, complete linkage. There is another associated term once we are discussing the complete and incomplete linkage, that is the linkage group. We shall also take account of it. Now, let us try to understand what do you mean by the linkage. In the first chapter, you have studied the Mendel's laws of inheritance, and one of the law was law of independent assortment. In the law of independent assortment, Mendel described that, say for example, here you are having the two pair of genes. One pair of gene is for the shape of the or the color of the seed, and other is the for the shape of the seed, yellow color and R is round, round is shape. So, you are having the dominant pair of the genes, and on the other hand you are having the wrinkled and the green seeds, which are recessive in characteristic feature. So, I need not to discuss with you the dominant, recessive, because all these things you have discussed, you have studied in the first chapter. Now, according to law of independent assortment, what will happen, that it is not always that the gene Y for the yellow will always remain with the gene for round, that is R, and accordingly the gene for the wrinkled small y shall not always remain with the green, that is small r, from one generation to other. Say for example, when both of these were crossed in the F1 generation, though the phenotype was again round and yellow, because here the dominant Y and dominant R are present, but it is heterozygous and it is showing its morphological or the phenotype with one of the dominant parent. Now, when you look at in the F2 generation, then you see that when you go for this crossing over in the F2 generation, for the production of the F2 generation, when you are crossing the F1, one F1 with other F1. So, you know, four possible types of gametes are formed, again I will not discuss them, but what was the law of independent assortment? You can see over there that sometimes this yellow gene is with the R, yellow gene is with the round gene, sometime this yellow gene is with the wrinkle gene, you can see over there small r, sometime the green gene is with the round gene, and sometime there is a this green gene is again with the wrinkle gene. So, you are having four different types of combinations, the round and yellow, round and green, wrinkled and yellow, and wrinkled and green. So, if you look at, they were the two major characters, round, yellow, wrinkled and green. So, it was not always that round was always found with the yellow, sometime round was found with the green also, and sometime wrinkled was found with the yellow also, though it was originally in the parent, wrinkled was with the green. So, it means here the genes have separated themselves in the generation independently, they are not dependent upon one another. So, it is because of this reason that round has gone with the green also and the wrinkled has gone with the yellow also. So, this was the law of independent assortment by the Mendel, but in fact it is not always that you will find this condition, which as I have explained you in the law of independent assortment. So, this law is not universal. This law holds good only for the characters when the genes are present on the same chromosome, the genes are present on the different chromosome. So, they will go separately only when the R will be present on the separate chromosome and the yellow will be present on the separate chromosome, because in the meiosis and meiosis you have studied that the chromosome go in the next generation as a unit and all the genes present on the chromosome will go together, and this independency is possible only when the genes, say for example, in the case of this Mendel, both the genes were basically present on the two different chromosomes. Okay. So, he came to this conclusion, and if they will be present on the same chromosome, then the law of independent assortment may not be visible. So, it means you know that it is always that the number of genes are more than the number of the chromosomes. So, it means we cannot say that one chromosome is holding one gene. So, it means on a chromosome you may have two genes, more than two genes, so many genes, because the number of genes are always more than the number of the chromosome in a body or in an individual. So, independent assortment is possible only when both the genes are located on the different chromosome, but when both the genes are present on the same chromosome, they will go together. So, in that case you may not get themselves, you may not be able to observe themselves in the generation in a separated condition. They will always be they will always go together, since the chromosome move as a unit. So, once this chromosome whole will move as a unit, then the gene 1 and gene 2 will go together. They will not be separated as explained by the Mendel in the law of independent assortment. Now, it means this tendency of the gene, when they are present on the same chromosome, that they will stay together, they will not get separated during the course of hereditary, that is transfer from parents to offspring. And once the genes are present on the same chromosome and when they will stay together, then they form the linkage. That means they are linked with one another. The genes located in the same chromosome are called the linked genes. So, in the previous example, gene A and gene B were linked together. This example can be found in the Drosophila melanogaster, in which gene for the red eye color and the gene for the gray body always found together in the offsprings. So, because both the genes are present on the same chromosome and they are the linked genes. Then the principle of linkage was first discovered by Bateson and Punnett in 1906. However, it was put forward as a regular concept by Morgan in 1910. Now, based upon the observation that when the genes are present on the same chromosome, they will go together, they will show some sort of relationship, they are called the linked genes. Morgan and Kessel formulated a chromosome theory of linkage, because here the linkage is with respect to the chromosome. That is, gene lie in a linear order on a chromosome. Say for example, this is a chromosome. So, one gene is there, second will be there, third will be there, they are not overlapping, they are present in a linear fashion in a longitudinal axis. The gene which show linkage, that means those genes which are present on the same chromosome, they will stay together in a hereditary transmission, that when the parents will form the gamete, this chromosome carrying both the genes A, B will go to the same individual. So, say for example, here also you can see in the gamete that this capital A and capital B here and this small and small b here are present over there. So, if they stay together, so they can be called as the linked genes. So, it means wherever this chromosome will move at the same location, both these genes will go. So, genes which show linkage, that is stay together in hereditary transmission, are located on the same chromosome. So, if you are finding that some characteristics feature, as we have discussed in the previous slide with respect to the Drosophila melanogaster, when both the characters were together, always found together in generation to generation, it means you can say that both the genes are the linked genes, they are present on the same chromosome. Genes tends to stay in parental combination except for occasional crossing over. So, if the genes are present in a linked fashion, even then sometime they have the chance, as you can observe in these two chromosomes, that capital A has gone with the small b. It is because of the crossing over which you might have studied during the course of meiosis. So, when the crossing over takes place by exchanging the segment of the chromosome, the one gene may go with other and other may go with the first one. So, here the capital B has gone to this after separation. Once after crossing over, chromosome will separate and the small b has come with the this one along with the capital A. So, even then if the genes are present on the same chromosome, they may have the chance to get themselves separated because of the linkage. So, the distance between the linked genes in a chromosome determine the strength of linkage, means if the genes are located very close together, they have less chance to get separate during the crossing over. If they are widely apart, they have more chance to get themselves separated during the crossing over, because say for example, if A will be located over there and the B will be there, then even after the exchange of the segment you will find that the A B will remain together. But since A is present on the other side, B is present on the other side, middle segment has crossed itself. So, it has exchanged this part, you can observe over there. So, if the distance is more, there are more chances of the crossing over and more chances of the formation of the recombinant. If the genes are located very close together, there are more chances that in the gamete formation you will have the parental combination. This is the parental combination because it is exactly similar to the parents, and this is the recombinant where they have exchanged their parts. Now, you are having two kinds of linkages. Number one is complete linkage and other one is incomplete. Let us look at the complete linkage. The genes which are closely located in the chromosomes, they will show the complete linkage, because as I have already told you, they have no chance of separating by crossing over and are always transmitted together to the same gamete, and then they will be observable in the same offspring. Thus, the parental combination of trait is inherited as such by the young one in case of the complete linkage that we have already discussed in the previous slide. Let us take an example over here. This is the red eye normal winged Drosophila when it was crossed with the purple eye vestigial wings. Note down that the genes for them are present in the homozygous condition. So, they are pure. Say for example, for the red eye it was capital R and for the normal wing it was capital W. So, it was pure dominant and it was pure recessive because both are the small. Now, you know that two types of gametes may be formed. One will, this R may go with this W. So, again this W may go with this R, again same. So, you may have the capital R and capital W type of gamete, because the gamete are haploid, they will have only one gene of the, they will have only one chromosome from the homologous pair. So, again say small r and small w. When they are crossed in F1 generation, so the F1 genotype was having this type of combination. Say for example, there are chances that this RW, the capital R and capital W may go with the small r with the small w. So, same here. So, you have the again, because the R is capital, W is capital, it is dominant character. The phenotype is red eye and normal wing in both the cases, but it is hybrid, it is not pure, because in the pair both the genes must be of the same type, but here the capital and small both are put together. Now, the F1 flies are all heterozygous, that is they are not pure, that is they are all are red eyed with normal wings. Now, in order to check that whether this F1 is pure or not, you can conduct a test cross. In the test cross, what you do, you will take this red eyed and normal winged F1 hybrid which was produced over there, and you will cross it with the pure recessive parent. When you will cross it with the pure recessive parent, then in the you can see the two types of gametes are there, capital R, capital W, small r, small w, but here only one type of gamete, that is R, small r and small w. Then this capital R and capital W may go with the small r, small w. So, this may be the condition, and again this may go with this, and this may be the condition, and when this small r and small w of this will go with this, then it will form the both the having the you can say the recessive characters. So, here when the individuals were found after crossing, that the red eyed and the normal winged and the purple eye with the vestigial wings were produced exactly in 50:50 ratio, 2:2 or 1:1 or 50:50, whatever you can say. Now, you can see no recombinant type are found, that means red eye and normal wing again it was parental, purple eye and vestigial wing was again the parental, no new combination is formed over there. It is not so that the red eye may have gone sometime with the vestigial wing or the purple eye have gone with the normal wing. No, both the F2 generations were showing exactly in the test class 50:50 ratio with respect to the parents. Now, it means this is the complete linkage. Complete linkage means both the genes of these two characters are so closely placed in a single chromosome that even after crossing they are not getting themselves separate. So, this linkage is called the complete linkage. Let us look at now incomplete linkage. So, say for example, linkage is there because the genes are present on the same chromosome, but they are present widely apart. So, they may show the incomplete linkage. Let us see what will happen in them, because they have the chance of crossing over and getting the two segments separated. So, capital A may go with the B, small b and small a with the capital B. So, there may be a recombinate. Now, here you can see it is not 50:50 ratio. So, you are having the parental combination and you are having the recombinant also in the 50:50. Earlier it was both the parental combination was 50:50 ratio. Let us have a again cross between double dominant, again it is double dominant because both the genes are capital with the double recessive, blue flower and long pollen. It is of the pea plant, sweet pea plant, and red flower with round pollen. This is recessive, this is dominant. Two types of gamete may be formed, BL and BL, again here small b small l, and in the F1 this capital B may go with the small b and capital L may go with the small l, and again you may have the same type of condition over there. So, what is over there, that again in the F1 you are having the blue flower and the long pollen, blue flower and the long pollen, because in both of them the dominant character is there. But note it down, in the previous one again we were having the F1 generation exactly with the dominant parent, but here again you are having the F1 generation exactly resembling with the dominant parent, but in the test cross it will show a different result. What will happen over there, that when you are conducting the test cross between your F1 generation heterozygous F1 generation with the dominant recessive parent, this one you are having few more combinations which are called the recombinants. That is, now over here blue flower, long pollen, this was the parental combination, round flower, round pollen was the parental combination, but two new combinations are there, that is blue flower with round pollen and red flower with the long pollen. Now, look at the ratio over there. Ratio is 7 this parental dominant, 7 parental recessive, 1 parent one the recombinant one type and 1 recombinant other type. So, what do you mean by this? This is not exactly 50:50 ratio. It is not 50 ratio of the parental with respect to the 50 ratio of the recombinant. It is having less recombination and more parental combination. That means, otherwise these genes were linked, they were linked genes, they were showing the linkage, but due to crossing over, some of the chromosomes have crossed themselves. So, there is a very less proportionately very less recombinant individual present in the F2 generation. Had it been so that the linkage was complete, if there was no chance of their separation, then both the individuals were having the exactly parental combination into 50:50, or even if it was having the this new recombinant, it would have shown exactly 1:1:1:1, but instead of showing this normal dihybrid cross, it is showing unexpected 7:1:1:7. This means the dihybrid test cross involving independent assortment of 2 genes, the frequency of 4 phenotype is 25%. That means, as per Mendel's law, if all the genes were showing the independent assortment, then the new recombination must be like this 1:1:1. But here you have seen that parental combination is that means parental combination is not similar with that of the recombinant, that is parental combination is more, which is 87.4% as compared to the recombinant, which is 12.6%. So, it means the genes were linked, they have not they were not having a full chance of separation always, but they have get themselves separated into a very few recombinants because of the crossing over, otherwise they were linked. So, it means whenever you are getting less than 50% frequency of recombination in the dihybrid cross, that means dihybrid cross, when you are testing it, you are not having the 50% recombination, it is less than 50% recombinant, it means it is a incomplete linkage. Linkage is not complete, linkage is not strong enough, sometimes the genes are getting separate, and I have already explained you, it is because of the reason that the genes are present on the on a widely separated part of the chromosome. Though they are present on the same chromosome, they are linked, but they have the chance because they are widely apart from one another. Then this was a linkage group, as I have already told you that all the genes in a particular chromosome form a linkage group. Say for example, if these are the genes present on a chromosome, one chromosome, they are the genes present on the other chromosome, they are called they form a linkage group. So, in this you are having 2 chromosomes, the 3 genes, and these are the alleles, because the homologous pair of the chromosome will have same gene present on the opposite side. Same means meant for the same character, though it is recessive, though it is dominant, but they are meant for the same character. Say for example, if this gene is meant for the color of the eye, it is again for the color of eye. The difference is only in the dominant recessive, it may be black, it may be white, but again it is same because both of them are denoting the color of the eye, say for example. So, a homologous pair is having the same type of allele. So, both the homologous pair, both the chromosome, the genes of both the chromosome of a homologous pair form a single linkage group. They are not the 2 linkage group, because both the chromosome are homologous having the same type of gene. This was a small concept with respect to the linkage group, which is not otherwise present in your syllabus. I just have introduced you. Then look at the factors affecting the linkage. One of the factor that we have discussed in the previous slide is the distance. That means, if the distance is more, you may have the chance to get the linkage broken. If the distance is less, then it will be a complete linkage, it will not show the new recombinant in the F2 generations. The due to crossing over, gene will not get themselves separate. In addition to the distance, age is one of the factor, that when the individual is having more age, then in the more age you have the less chance of the crossing over during the meiosis. Hence, the linkage is strong enough, the strength of the linkage is more with advancing age. That means, more old will be the individual, less are the chance of the crossing over and the less are the chances of the breaking of the linkage. So, otherwise this crossing over is very important, because whenever there is a crossing over, when there is a new combination of the gene due to crossing over, then variants are produced, and when the variants are produced, variants are always more vigor as compared to the non variants. So, always variation is required for a good individual for the evolution to occur. So, it is because of this reason that the doctors often prescribe that the male and female must get themselves married not in a well suitable age, not in the late age, because in the late age you may not have the good chance of the crossing over. As a result, your parents, your offspring will not be having the variations, and once the variation is not there, that means in the it is not a good sign, because variation is good for the evolution to occur, variation is good for the individual for its strength and the vigor. Other than that, temperature also affect the linkage. When the temperature is increased, then there are chances that the chromosome may get broken. As a result, the linkage will not be having a good strength, it will be a weak linkage, and it will show a incomplete type of the linkage. Exposure to the X-rays again reduce the strength of the linkage, because they have the chance to get the DNA broken. As a result, the chromosome may be broken and the genes may be separated, which were otherwise having the linkage system, they were the linked genes. Now, what is the significance of this linkage? It greatly reduces the chance of formation of new combination of genes in the gamete. So, if the genes are linked, it means new combinations will not be formed in the genes. It thus help keeping the parental, racial and specific trait together. So, once the variation will not takes place, then the parental character, then the character of a race, then the character of a species will remain same from one generation to other. So, this is one of the benefit of the the significance of the linked gene. Second significance is that it helps in maintaining the valuable trait of a newly developed variety. Say for example, if the scientists are successful after crossing in putting the genes present on different chromosome to a single chromosome by recombining the genes, then if both the genes are present on the same chromosome and they are linked enough, it means the characters that they have developed after combining the genes present on the different chromosome because of the new combination of the genes will not get be destroyed in the future, because the genes present on the same chromosome will always go together. In order to understand it, you have an example. Say for example, in your maize crop you are having a long plant with a good corn size and a small plant with a very small corn size. Now, because long plant is having an advantage of good corn size, but having a disadvantage of going wasted, broken when wind will occur. So, on the other hand, small plant will not be broken because of the wind, but the corn size is small. So, again it is not economical for you. So, scientists have tried that let us put the gene of good corn size present in the long plant to the gene of to the on a chromosome along with the gene of the small size plant. So, it means they are linking the size of the plant with the size of the corn, that both the genes must stay together. Whenever there must be a small size, it must have a good corn size, otherwise it was not so. So, if the scientist will remain successful in putting both of these genes to a single chromosome, they may be able to develop the linkage group or the linked genes. Then the character that they have developed, that the small plant will have a good corn size, will remain together in generation to generation only because of the linked genes or the linkage concept, and that too because of the complete linkage concept. Linkage disallows the plants and animal breeders to combine all the desirable traits in a single variety. So, because so many genes are linked together, they are showing the complete linkage. So, it is not always that when you cross one variety with the other, you will not always get a desirable trait. So, this is a restriction upon by the nature upon the scientist. So, this is another significance of the linkage. The fact that there are as many linkage group as the chromosome pair in is an important evidence for the location of gene in a chromosome. It is a very technical. Say for example, you are looking at some certain characters that they are visible in the individual always together. Say for example, as we have already discussed in the previous slide, I am taking another example. Say for example, in an individual you are looking at whenever say for example, in the maize also, whenever there is a short plant and the short plant has the low quality of the corn. So, if you are able to detect a gene either of the two, either of the for the size or for the size of the plant or for the size of the corn, if you are able to locate that this chromosome is having gene for this size of the plant, and you are seeing it that always size of the plant is related with the corn size. So, it means you can immediately say that the second gene is also located on the same chromosome. So, it is how that it helps in location of the genes in a chromosome.